everyone. Thank you for joining us. Welcome to the Society for Marine Mammalogy's monthly Editor's Select webinar series, where we highlight the latest exciting research in the Marine Mammal Science journal. Thank you all for being here today. My name is Teresa. I'll be hosting today's webinar. I'm one of the student members at large for the Society for Marine Mammalogy, and I'm also a PhD candidate at the University of California, Santa Cruz. So this month, we're really thrilled to welcome Dr. Solène Derville, who'll be telling us about her research on the behavioral response of humpback whales to biopsy sampling in New Caledonia. Solène has a master's in biology from the École Normale Supérieure in Lyon, France, and a PhD from the Sorbonne University in Paris. And she's currently a postdoc in the Geospatial Ecology of Marine Megafauna Lab at Oregon State University in their Marine Mammal Institute. She lives and works in New Caledonia, however, where her research focuses on animal behavior and spatial ecology, and she's really interested in developing innovative and multidisciplinary measures and methods to study marine megafauna species. So as a reminder, we are also live on Facebook. This recording will also be available on YouTube at the end of the talk. And there'll be time for questions at the end of the presentation. So we ask that you please use the Q&A function, which you can find at the bottom of your Zoom screen, to send in any questions. The paper that this is based on is also available open access through the Marine Mammal Science Journal website. And so with that, I'll turn it over to Solène.
Okay, I'll unmute myself. Thank you. Thank you for the introduction. Really happy to be presenting this work today. So I'll start with this photo that was taken a couple years ago in the fields during one of our research cruises. So this is my former PhD supervisor, Claire Garrigue, and myself processing some humpback whale samples that we just collected as part of our monitoring of the humpback whale population in New Caledonia. And this photo really illustrates the result of a good day at sea, a successful day in the field where we've collected a lot of samples. This photo could have been taken anywhere in the world from any place where people study cetaceans. But my talk today is going to be about what happens before we happily fill that little bright orange box where all of our samples are stored. Because we have this responsibility to assess the impacts and the disturbance that might occur as a result of our research activities. And that's what we've tried to do in this study with Claire. So my talk today is about the response of humpback whales to biopsy sampling, and I'm really happy to be presenting it on behalf of Claire and myself for this SMM Seminars Editor Select series. So before I start, I would like to give a big shout out to Claire, who's been my mentor for many years. Claire is a senior researcher at the French National Research Institute for Sustainable Development in New Caledonia. She is also the founder of a local NGO called Opération Cétacé that has been conducting research in the region for over 25 years. Claire was my PhD supervisor, and although I'm now a postdoc at Oregon State University in the GEM lab, I still have a few side projects left over with Claire, and among those projects is this study that we just published this year in Marine Mammal Science about the impacts of biopsy sampling on humpbacks. So for those of you who don't know where New Caledonia is, that's totally understandable given how small this island is. It's a French overseas territory in the Southwest Pacific. Although it's a small island, it has a very big economic exclusive zone, which I've represented here on this map. So New Caledonia is composed of a mainland with a very large tropical lagoon surrounded by a very long barrier reef. But it also has different remote reefs, such as the Chesterfield reefs that are located halfway between New Caledonia and Australia, and the Loyalty Islands to the east. So the NGO Opération Cétacé was created in 1995 to conduct research and conservation of marine mammals in this region in the South Pacific, and most specifically in New Caledonia. So the NGO has been monitoring the New Caledonian humpback whale subpopulation for over 20 years. And although humpbacks have been studied a little bit all over the EEZ, most of the work has focused on the South Lagoon, which I've circled in red here. And as you can see, this is a pretty nice place to work in, as you can judge from the photos that I've put at the bottom of this slide. As part of our everyday field routine, we've been collecting biopsy samples of humpback whales for over 2 decades in New Caledonia. But why do we need these tissue samples? Tissue samples of cetaceans can be used for many different types of analysis, including, of course, genetics, which includes genetic aging or genetic sexing, but also isotopes analysis, analysis of pollutants, of hormones, and even microbiomes. And these analyses help us answer many ecological, biological, and behavioral questions that eventually can inform conservation of cetaceans. How do we perform these tissue biopsy samplings? Well, for large whales, the most common approach is to use a crossbow or a rifle. The crossbow is definitely what we've been using the most in New Caledonia. So this is a photo of Claire taking a remote biopsy sample with a crossbow on a humpback. And for those of you who might never have witnessed a biopsy sampling, I just wanted to show you a short video. So this is me on a small dinghy taking a biopsy. So there's a humpback to the right here that I'm pointing at too. We're taking photos at the same time, but that's not actually the one we're going to biopsy, because ta-da, there's one to the left, and there we go. So the arrow is just floating at the surface, and now we're going to circle back with the boat and collect that arrow and collect the tissue sample that is in the tip of the arrow. Okay, so the other device that people can use to do remote biopsy sampling is a rifle, a veterinary rifle. And this is most commonly used for small cetaceans. So here you have a photo of dolphins being biopsied with a veterinary rifle. And this little dart here is what is going to collect the tissue sample. There's a further approach that can be used to collect skin samples, and that I wanted to mention today. It's not really the topic of this talk, but I think it's important to mention it. For certain species of cetacean, it is possible to skim the surface of the water to collect slough skin. So humpback whales, but also other species, tend to lose slough skin, to shed it at the surface of the water when they perform very intense activities. And that's a good opportunity to collect samples. However, we must note that it's not possible to collect blubber in that way. And it is sometimes a little hard to identify in the field which individual has been performing activities and to associate the tissue sample with a specific individual. So the slough skin is not really used in the same way as the tissue samples that are collected with a crossbow or a rifle, but it's important to note that it's still interesting way to collect samples in a totally non-invasive way. So what is the impact of this tissue biopsy sampling, and most specifically the remote tissue biopsy sampling with a crossbow or a rifle? Of course, the long-term impact of tissue biopsy sampling, but actually of any kind of anthropogenic activity, is really hard to estimate and is unknown to a great extent. So one possible way to assess disturbance or impact is to look at a short-term behavioral response. And this is what other studies have done. So short-term behavioral impact have been assessed qualitatively in the field in different breeding grounds for humpback whales, mainly in the Atlantic. These studies have found that there was an effect of the social context, the reproductive state, and the age class of the animals that were being biopsied. So here I've reproduced a figure taken from Claham and Mathill in 1993 that shows the reaction of different classes of humpback whales to the biopsy sampling. They've classified the reactions into 3 categories: no reaction in black, low reaction in gray, and moderate reactions in white. And here I've highlighted specifically the responses of the calf categories, so the young of the year. Claham and Mathill showed that calves had stronger reactions to biopsies than all other adults that they've tested in this analysis. However, there were few replicates of that study because calves are not routinely biopsied as much as adults, and there were places where research permits and regulations do not allow for the biopsy of calves. So this takes me to all of the questions Claire and I had about the whale response to research activities and how this response could be influenced by different factors. So we wanted to look at intrinsic factors such as age class, reproductive status, and repeated sampling, but also at extrinsic factor like social context, sampling device, and habitat. Okay, so I've already presented our study sites a little bit. This map on the bottom left is showing all of the biopsy samples that were taken by our team around New Caledonia. So they're shown with little orange dots. As you can see, there's a few in the Chesterfield, a few around the mainland and in the Loyalty Islands, but most of the samples come from the South Lagoon, which is the historical breeding grounds that has been monitored in New Caledonia, and quite a bit in what we called the Southern Seamount. So this is a specificity of our study region where we've been working quite a bit in some pelagic environments, some seamounts that are located south of the mainland and where whales tend to aggregate in large numbers. So our data set covers more than 2 decades, from 1995 to 2019. And the remote biopsy samples have been indifferently collected from adults, juveniles, and calf humpback whales. It's also important to note that this subpopulation that comes to breed and calf in New Caledonia is part of the endangered Oceania stock. So we were also interested in what the response of these animals would be in the context of an endangered population with relatively low densities compared to other breeding grounds around the world. So the first variable we looked at was the response to the boat. So in the field, we'd always try to assess if the whale actively avoided the research vessel during the approach. This resulted in a very simple binomial variable that said, yes or no, did the whale react? It's important to note that this response was only recorded for the time during which we are making the attempt to go biopsy the whale, and not throughout the entire focal follow, which can make a difference in the end in terms of frequency of response to the boat or not. I'll come back to that later. Then we looked at the response to the biopsy sampling itself. So we looked at the reaction of the whale right after the arrow or the dart hit its back. And this response was qualitative. It was ranked from no response to weak to moderate and strong response. And this ranking was based on a definition given by Wenrich et al. In their work from the 90s, where they've defined what a low, moderate, or strong response could be. So a low response here is a brief and mild response, something like a startle or an immediate dive. A moderate response is more forceful, but it is not prolonged. So it would be, for instance, just one tail slap or one tail flick. A strong response, however, is both forceful and prolonged. So it would be, for instance, having multiple tail slap in a row. Now we looked at these two variables, the response to the boat and the response to the biopsy, as a function of many different factors, as I said, both intrinsic and extrinsic factor. The main thing we were interested in initially was age class. We were very curious to see if cows would have a different response from adults. But then we realized there was also a third category that we could differentiate in our data set and in the field, which was the category of juveniles. So juveniles are a little hard to identify in the field. We have to acknowledge that. But in a breeding ground, they can be identified from their small size. They're not as small as calves. They're more than 1 year old, but they're not as big as a mature adult. And they can also be identified at the coloring of their fluke, which is a bit blurry. And I'll show some pictures later to show you what I mean by blurry patterns on the fluke. Then we looked at the effect of sex and reproductive status. So we looked at the different response between males, non-lactating females, and mothers. And it is to be noted that these, the sex was genetically identified in all of these cases, except when a mother was seen with a calf and then it was identified as a female. Okay, social context was a little more complex. For those of you who don't know about humpback whale breeding systems, they tend to form small groups that usually associate for a short period of time. So the females, either with or without a calf, could be found either alone or a singleton, in a pair, or in the case of a female with calf, we call this an escort. It's a male escort that joins the mother and its calf, or they could be found in competitive group. So a competitive group is when multiple males start chasing a female to gain access to it. These groups can go up to about 10 individuals in New Caledonia, maybe even more in other breeding grounds around the world. And these groups usually have very intense surface activity and high speed. For males, the story was a little more complicated. Again, we identified 3 different social contexts, whether they were alone, in a pair, or in a competitive group. And then within the competitive group, we were sometimes able to identify what the role of the individual exactly was. So we've differentiated the main escort, which is the male that is closer to the female, the secondary escort, and the challengers that are trying to take the place of the main escort. In terms of the methodology used in the field, we were super interested in how whales would react with a biopsy taken with a rifle compared with a crossbow. So we also made that comparison both for adults and for cows. Then we looked at habitat. So as I was telling you earlier, in New Caledonia, we work a lot in a near-shore breeding ground called the South Lagoon, which I've shown here on the map. But we also work a lot on some southern seamounts that are located south of the mainland. So these seamounts are Torchbank, which is about 30 meters deep at the summit, and Antigona Seamount, which is about 50, 60 meters at the summit, with the surrounding waters being about 1,000 to 1,500 meters deep. So this is a very different environment. It's offshore, it's totally unsheltered. And at some times of the year, well, of the season, the whales aggregate in rather large numbers and high densities over these, the summits of the seamounts. So we thought this could also affect the behavior of the animals and therefore their response to the biopsies. Finally, because we had such a long data set, we could look at the effect of repeated sampling, which was a pretty novel analysis in terms of the impact on whales of the biopsy sampling. So first, we looked at within season repeated sampling. So of note here, of course, we are trying as much as possible not to biopsy the same whale multiple times throughout the season, because that would just be a duplicate of the sample. But because we have a small population, it happens just by chance that sometimes we biopsy the same whale twice or 3 times within the season. And those events were used here to assess within season potential increase or decrease in the intensity of the animal's response. So we looked at the intensity of the response to the biopsy as a function of the repeats, the social context at the time of the biopsy, and we used a mixed effects type of model that would account for individual variability as well. We then looked at repeated sampling across seasons, and this time we had many more samples because, again, we have a small population with a strong reciting, with a higher reciting rate, and therefore we could see if the response to biopsy changed throughout the years from 1995 to 2019, again running that analysis at individual level, and also including as a cofactor the time that elapsed since the last biopsy happened. Okay, so moving on to the results now. We recorded 2,288 responses to the boat, and in 95% of the cases, so the great majority, we found that the whales did not react to the boat approach. So this result is pretty strong. However, it must be noted again that this response is only recorded during the biopsy attempt. So it does not necessarily reflect the behavior of the animal throughout the whole focal follow. And I bet that if we had measured that avoidance throughout the whole follow, then we might have had a bit more responses to the boat. Then we recorded 2,248 responses to the biopsies. And as you can see, we also had a majority of cases where the whales did not respond to the biopsy. That's 60%, which tends to be a little more than what other authors have observed in humpback whale feeding grounds. We also had quite a few weak responses, 17%, some moderate responses, 23%, and only very, very few strong responses, 0.5%. So as a reminder, the strong responses are the ones where the animal is repeating the same reactions several times in a row, and it looks like this really rarely happens with humpback whales. In addition to qualitatively ranking the response, we also took notes of any kind of specific activity that the whale had at the surface following the biopsy. And we found that in 78% of the moderate or strong response cases, the whales were performing a tail flick. So this really looked like the usual response that the humpback whale might have to a biopsy sampling. Okay, so as I said, age class was really our main interest when we initiated that study. And we were really interested to see that adults and calves did not significantly differ in their response to the biopsy. So this is shown on this figure that is taken from the paper, where you have the proportion of responses on the y-axis and the different age classes on the x-axis. The numbers within each of the boxes here are the sample size within that category. And as you can see, the calves and the adult had pretty much the same response rate. Juveniles, however, had more intense response than adults and calves to the biopsy. So this result is similar to that found by Best et al. 2005, where they've studied the response of southern right whales to biopsy sampling. They did not find a difference between calves and adults' response. And the explanation here, we think, is that calves have a lot of physical contacts with other whales during the breeding season, and they might just be used to being touched or hit by something at any time during the day. So here I've pasted 2 photos at the bottom that show cows in pretty violent, intense interactions with males in competitive groups. So on the left, there's a male almost breaching on top of a calf. And to the right is a picture of a calf being lifted out of the water by a male during a competitive interaction. So as you can see, yeah, life as a calf is not really easy. But they also have a lot of close contact with their mother. And to illustrate that, there's this really nice picture at the top here where a calf was just playing on the back of its mother. And we think that those contacts as well might create an habituation and attenuation of the response to anything that would hit or touch the calf. However, so we were surprised to see that the juveniles reacted the most. And we think that's because juveniles are often found alone. They're less social than other adults, and therefore they're less subject to repeated physical contact. So this was a pretty novel result, although we have to acknowledge that identifying juveniles in the field is quite subjective and really hard. I mentioned earlier the blurry patterns and coloring of the underside of their fluke, and I just wanted to provide a few examples of this blurry pattern with these 3 photos taken in New Caledonia of juveniles, and you can see this grayish black pattern that we know tends to go away with time. And so we've used that also as a characteristic in addition to small size to identify juveniles. Okay, so the other things we looked at in the case of calves was if there was any difference between males and females, or if there was a difference in response to the rifle or the crossbow, and we found no difference, which is a pretty important result in terms of methodology in the field to know that rifle and crossbow apparently did not generate more reaction from the calves. For adults, we looked at habitats with the South Lagoon versus the seamounts, and rifle versus the crossbow. And again, we found no difference in the response to the boat or to the biopsy. The sex and reproductive status did have an influence on the response of animals. We found that mothers responded more to the boats than other groups, but had the least responses to the biopsy itself. The social context did not appear to influence the response of mothers and non-lactating females. So we found that mothers were the least sensitive to biopsy, which is a similar result to what was found in other studies in breeding grounds in the North and the South Atlantic. What we saw in our study was actually really reflected well what we observed in the field, that mothers have this kind of dual response. Either they clearly avoid the boat, and sometimes they won't even let you get close enough to get a biopsy. And other times they are totally oblivious to both the approach and the biopsy itself. So it's really interesting to see that they have this, this dual response to the boat and to the biopsy. Now, continuing into the effect of social contexts, among males, we did find an effect whereby males had less intense response to the boat and the biopsy when they were within competitive groups. Then looking at the role within the group, we did not see a difference depending on whether the male was a challenger, secondary escort, or a principal escort. So it's not that surprising that males would respond less to biopsy when they were within a competitive group. It's consistent with other impact studies. And we hypothesize that this lack of reaction is due to the violent physical contacts that males experience within the competitive groups. So these groups can be pretty intense. Males get injured, notably because of the barnacles that humpback whales have on their flipper and their chin. So I've shown them here on these 2 pictures. And possibly a biopsy sampling is probably not much in comparison of these competitive interactions, which would explain the response of these animals within those groups. Now, moving on to the final factor that we looked at within this study, which is repeated sampling. So first, we looked at the effect of repetition of the sampling within season. So for that analysis, we had 266 individual adult whales. So I would like to note here that we removed juveniles and calves to run these repeated sampling analysis. And this totaled 578 biopsies with a maximum number of 5 biopsies per individual per season. So the plot to the left here shows the proportion of reaction types. Again, no reaction, weak reactions, or moderate and strong, which were grouped together. And we look at this as a function of the order of the biopsy event for the season. The numbers at the top indicate the sample size. So we had 266 individuals that had been biopsied twice, 39 that have been biopsied 3 times in the season, and 6 that have been biopsied 4, and just only 1 that had been biopsied 5 times. We found that there was no significant change through time within a season in the response of these animals. Across seasons, the story was a bit different. In that case, we had 387 individual adult whales, which were biopsied 1,183 times, with a maximum number of 18 biopsies for one individual. Again, that plot is the same as before, the proportion of reaction on the y-axis and the order of the biopsy events from 1995 to 2019 on the x-axis, sorry. And in that case, we did find a significant attenuation, a significant decrease in the responses of whales with repeated sampling across seasons. So why does the response to biopsy decrease across seasons at individual level? We have different hypotheses to try to explain that result. First, this can be a methodological bias that could be related to shifting baselines in the perception of the intensity of the response. It is possible that observers have been ranking responses at a lower level of intensity in the most recent years. The second possibility is that there was a change in the field method that might explain this decrease in the response. And actually, we did realize that in the recent years of this work, the photo identification tended to be performed before the biopsy sampling when the team was in the field. Whereas in most recent years, we tended to perform the biopsy first. And we think that the prolonged focal follow with the animals, so doing the photo ID and then the biopsy, might have made the whales more annoyed with the bow presence, and that could cause the reactions to be more intense in those earlier years of the study. It's also possible that the responses have attenuated due to age, because of course, throughout these years, the whales have grown older, and that might have affected their baseline stress level or other behavioral aspect that made them less sensitive to the biopsy sampling. And finally, it is possible that there is an habituation to the biopsy sampling itself. So it's important to note that we found no sign of site avoidance or reduced sighting rates, resighting rates for time. We looked at this using goodness of fit tests for trap dependence and the probability of recapture. And this is just important to note because it shows that there is no major population level avoidance of the study site that would be due to our research activities or to any other human activity that might be causing disturbance. So in terms of implications, I mean, first, I want to really acknowledge that this study was a little opportunistic. It's the result of more than 20 years of data collected very consistently, but in a qualitative way, and just looking at the short-term behavioral response of the animals. But further research should investigate the potential long-term effects of these research activities or other research activities, and using a more quantitative approach. I think this work really highlights the value of collecting long-term data set very consistently. This work could not have been performed without the huge efforts of Claire and her team in New Caledonia over so many years. So again, it really again justified the need for more long-term monitoring of cetacean populations and behavior and ecology. Finally, we hope that this work can have implication and can help managers setting up research authorization for depending on the social context, on the habitat, and on the type of animals that are targeted by research activities. Okay, and with that, I'm going to thank you for your attention. If you want to get in touch with us, please don't hesitate. I've pasted Claire's email address and mine on this slide. We also have a Facebook page to showcase all of the work that the NGO Opération Cétacé is conducting in New Caledonia, and Claire and I have Twitter accounts as well. Yes, and with that, thank you for your attention, and don't hesitate if you have any questions.
It was fascinating, Solène. Thank you. Yeah, so just to emphasize that again, please, those of you who are watching and listening, go ahead and post any questions you have in that Q&A section that you'll see at the bottom of your Zoom screen. And Eric will read those out for us as they come in. While we give people a moment to answer, I guess I had a question which you might be able to answer. I know you're studying, these large whales. Can you talk at all about similar research in smaller cetaceans and maybe smaller odontocetes? Do you know anything about that? And do you see similar lack of responses, or are there differences there?
Right, so there's a really good review that compiles all of this research conducted on the impact of biopsy sampling. That's Noren et al. And they included all cetaceans in there. So there's also odontocetes who seem to have stronger responses than large whales to biopsy sampling in general. Like those strong responses that I mentioned at some point that are repeated and forceful are very rarely seen in humpback whales, but a bit more in odontocetes and small dolphins and such.
Which I guess might make sense given all the factors that you discussed in terms of body size and interactions and all those tough barnacles and things like that. I don't see any questions quite yet. I encourage people to ask. I think it was just so clear and yeah, well presented that maybe folks don't have anything. I'll go ahead and ask another question that we always like to ask our presenters, which is separate from the research specifically, but if you can talk a little bit more about sort of like your background and how you came to, have awesome crossbow skills and be doing the research that you're doing, and related to that, if you have any advice for students, especially, as student members at large, we want to represent our students, advice for students who might want to get to somewhere like you are today.
Yeah, of course. Well, my background initially was in very general biological sciences and not necessarily marine biology and definitely not marine mammals. But I progressively specialized through internships and different research projects in different labs, started working on turtles and then on cetaceans. But the fact that I started working on humpback whales with Claire was luck, really. I was just super lucky to meet her and get to work with her. But in terms of advice to students, I think it's important to somehow find your niche, the thing that you like doing and you're good at. And in terms, I think, of methods rather than thinking in terms of species or habitats. So in my case, the research I presented here today is actually very different from the rest of the things I'm doing, which focuses on spatial ecology. And I think that's my niche for me. It's doing statistics and coding and mapping, basically. But for someone else, it may be genetics, or it may be isotopes or bioacoustics. But it's important to find a niche, I think.
That makes sense. Eric, do we have any questions?
Yeah, I've got a question here. This one's from John Moran. Any thoughts on reactions when you miss the whale?
Yeah, that's a really good question, because those cases of missed attempts were addressed in, I think, at least in Claham and Matilla's work, but we did not assess that within our study. It's something that we did record in our field books, but did not necessarily have well computed and consistently recorded in our database. So we ended up not using those cases. We also removed like all the cases where we did satellites, the deployment of satellite tags, obviously, because that's also a very different topic. But I agree, it's super interesting to see that a lot of the times whales react even if you don't, if the hit is missed. Just this, I guess, the sound of the arrow or the dart on the water next to them can generate a reaction, and that's definitely something we've seen in the field.
Thank you for that. Another question here from Natasha Pastor. In the US, we are required to ID the whales first to eliminate over-harassment of an individual. Do you have any country, federal, local laws that restrict the number of whales or individuals resampled within a season?
Right. No, we do not. It's not compulsory. We're trying to avoid it as much as possible, for sure. Like, I think other teams around the world use the same things, like printing the catalogs or having to get to know your whales better and be able to recognize them really fast once you're in the field, to try to not biopsy them twice. We're definitely trying to avoid that as much as possible.
Thank you for that. I had a question. I was curious, I think you touched on a little bit, but generally, how did you think like individual variability of just like individual animals made a difference? Because I've done some studies with dolphins and manatees, and like and just in knowing some of the animals and seeing them more, you just see like how much they vary in how different animals respond to different things. And I've seen animals habituate and sensitize. I've seen like some of them that get more startled and others that get less startled to stuff. And I couldn't really, do you think there was just a lot of, were you able to perceive how a lot of individual variability affected that, since you were seeing a lot of the same animals over and over?
Yeah, I mean, I agree with you. Certainly, there must be individual variability with maybe just different baseline stress levels and just different behaviors from one individual to the other. Claire would be in the better place to give you good examples of whales that were particularly hard to biopsy or particularly easy across the years. But I'm sure that's something that happens, but it's really hard to assess. It's what we tried to look at with the repeated sampling analysis, where we used a mixed effects model, where we at least accounted for some of that inter-individual variability. But the social context was also always extremely important, and that's why we kept it as a cofactor in the model throughout the analysis. So whether the social context or just the individual specificities and behavior, which one weights more, I couldn't tell.
Thank you. Okay, I have another question here from Susie Tierlink. Thank you for a great presentation. We have a biopsy sampling component to our research, and I was surprised to see a whale have a strong reaction to a drone collecting blow on one occasion, stronger than I had seen from biopsy. Have you heard of similar anecdotes?
Huh, interesting. Well, I know that in New Caledonia, we don't use drones. We haven't made that move yet. So I don't have any other ideas or comparisons, but at least here, you can't fly a drone over a whale legally unless you have a research permit for it. And people have started using drones, and I'm glad that there is some regulations around it, especially from whale watching activities and so on. But it's a good question. It'll have to be assessed in another study for sure.
I can mention just a drop on that I worked with Ocean Alliance a little bit, looking at whether the humpback whales were responding to blow sampling, and it was like relatively few animals responded. But what was interesting is that their responses were kind of like not similar to the same thing you get from a boat. And I saw the same thing in both dolphins and manatees, where they responded to a drone, but just very differently than they would respond to a boat. So I think, part of what a whale responding strongly to a drone would be, them perceiving something, in a very different way than like a biopsy. I think it actually would be, I don't think it's as impactful if they can't see it. So I don't think if you're flying like in front of their face, it's maybe an issue, but behind them, that doesn't really seem to be an issue. But I think it's just a different modality too. Like biopsy sampling is like, they're feeling something somewhere and perceiving something, but a drone is like an object, kind of like a boat, which they're hearing and perceiving from a distance, maybe. I don't know.
Who knows? It's hard not to speculate and try to interpret these behaviors, but be very objective about it. And that's what we need, more of these studies that assess in the most quantitative or qualitative way the response of the animals.
Okay, we've got one more question here from Andrea Ferreira Kosam. I can't see your name. I apologize. Have you heard of lethal responses to biopsy from infection, for example? And are there studies investigating the scarring process after a whale is biopsied?
A lethal, is that what I heard? Is that
correct?
Yeah,
lethal as in death.
Lethal. I've never heard of it for large whales, but I wouldn't speak for small cetaceans, honestly. I do not know, and I'd rather say this, that I don't know. But definitely from what I could see in Noren's et al. Review was that there were a few studies of the physiological impact of the biopsy sampling, that's for sure.
Thank you. We just had a comment from Shannon Barbara Meyer, who says, thank you for the great presentation. Thank you, Shannon. And then another comment here from anonymous attendee says, you might be interested in checking out the 1991 paper by Weinrich about humpback whale sampling in the North Atlantic, behavioral response of humpback whales in the Southern Gulf.
For sure, yeah.
And then another thank you for your great presentation. And that is it for questions. Yeah, that was the last question.
Okay, well.
Yeah, I think we'll, unless we have any last questions that folks want to bring into place. Do you want to tell us briefly what you're going to be doing next? You talked about some of your like spatial ecology work. Is that what you're working on right now?
Yes, absolutely. That biopsy work that we did with Claire was a bit of a side project. I work with Lee Torres in the Marine Mammal Institute at Oregon State University now, and I'm studying the distribution of rorqual whales on the Oregon coast to try to help reduce the entanglement risk in fishing gear in that region. So yeah, totally different kind of research. But we do need biopsies as well. So
Do you have anyone doing, so I use biopsy samples from dolphins for fatty acid analysis, and I know you had mentioned like stable isotopes and things like that. Do you have anyone looking at diet or using some of your samples to do that kind of work?
in New Caledonia?
Yeah.
Yes, we're also working with Susan Bingston Nash at Griffith University as part of the humpback whale sentinel project, trying to use humpbacks as sentinels of what's going on in the Southern Ocean. And a lot of the samples we've been collecting in the past years, and everything we're going to collect this year, actually, hopefully next week, starting next week, we'll go to that project that also includes diets and different metrics of health as a response of conditions in the Southern Ocean.
It's great to be able to collect a relatively non-invasive sample that you can use for so many different projects, and to know that it doesn't seem to be impacting the animals too much, at least from your research.
Yes, Claire has been sharing her data a lot and generating a lot of collaboration through that long-term research and data collection. And maybe I'll end with that. I think it is our responsibility when we collect this data that it is made available and stored and processed in the right way so that it can be used and reused as much as possible.
It's a great plug for that kind of collaborative work. It's amazing how much you can do when you share it that way. Yeah. Great. Well, thank you so much for your time. We really appreciate it. That was fascinating and really beautifully presented as well. Again, we invite folks to watch the recording, which will be available on both Facebook Live and on YouTube after this. We encourage you to go find Solène and Claire's paper and read more in even more detail and check out their other work. And we will not have a presentation next month in August because of the conference, but we should be back in September with something else. So keep a watch on your emails, and we hope that you'll think about joining us again in the future. And I think with that, we'll go ahead and wrap it up.
Thanks, Teresa. Thanks, everyone.
Thank you very much. Bye.
Bye.